Okay, these videos will cover drawings from chapter 11, Animal Physiology. It's important to note that these drawings are just drawings. They are not a comprehensive review. In other words, there are many things that you're going to need to know and are going to be tested on that are not going to be covered in these videos. Some of these drawings are required. I'm going to put a red star next to the drawings that are required. In other words, drawings that you might be asked to actually produce, draw, label, annotate, etc. And some are just good to know. In other words, help you to deepen your understanding on these topics, or drawings that you may be asked to like visualize, label, answer questions about, etc. These set of drawings are going to cover the chapters in 11.1. There are no drawings that you really need to be able to produce or know in chapter 11.1 on immunity. So I'm not going to start out with any of those. These are going to cover drawings from 11.2 on movement, 11.3 in the kidney, and 11.4 on sexual reproduction. Let's start off with a drawing of the human elbow. The human elbow involves 3 bones. Okay, one of them is the bone of the upper arm called the humerus. And the humerus at the end, okay, has a layer of cartilage, which is going to help absorb the shock a little bit and reduce the friction between the other bones. And then in the forearm, we've got 2 bones. Okay, one of them connects to your thumb, and that is called the radius. It also has cartilage present at the end. And the other one kind of connects to the back of our elbow here, and then to our pinky, and that is called the radius. Okay, that's not true. That's called the ulna. Okay, this is the radius, this is the ulna. The ulna, again, like these other bones, also has a layer of cartilage to absorb shock and prevent any kind of friction. These bones are held together with some ligaments, but we're not going to show those in our drawing. Okay, the elbow is an example of a synovial joint. Synovial joints are kind of cool. They have this synovial capsule, which holds a bunch of fluid, which is there to help reduce friction, provide nutrients, etc. On the downside, it decreases the range of motion. So we're not going to see as much motion with an elbow as we would with a ball and socket joint like our shoulder. So what's a synovial capsule? Well, it's got 2 parts. It's got a synovial membrane, and that synovial membrane is going to secrete the synovial fluid that will eventually fill this joint capsule. Okay, and then we have something called the joint capsule, and that's there to provide a little bit of stability. It's going to actually unite all of those connecting bones, and it's going to surround that joint. We'll go back and label those later. We've got 2 sets of muscles involved with the movement of the elbow, and one of them connects this humerus with the radius, and that is our bicep muscle. So the bicep muscle actually connects to the humerus all the way up here, okay, but we'll kind of show that it's like sitting nicely along that humerus, and it's going to attach to the radius. Okay, so big bicep muscle kind of attaches to the radius here via a tendon. Tendons attach muscle to bone, so in cases like right here, attaching a muscle to a bone. Ligaments, on the other hand, attach bone to bone, and again, we're not going to draw those in. Now, muscles always work in antagonistic pairs, okay, meaning that they're responsible for different ranges of movement. Since the bicep connects the humerus and the radius, when the bicep contracts, it's going to pull this radius up, resulting in a flexing of the elbow. The other part of this antagonistic pair, it also connects to the humerus, but on the back side, okay, and it connects to this part of the ulna, again, via a tendon. And this muscle is called the triceps. Okay, so when the bicep contracts, it moves the elbow up. When the tricep contracts, it's going to extend this elbow. All right, time to go in here and do some labeling. Again, this bone of the upper arm is called the humerus. Okay, this bone here is the radius. And then we have the ulna. The radius, the ulna, and the humerus all end in cartilage. Okay, and that again is there to absorb shock, reduce friction, etc. It's a synovial joint, so it is going to be filled with synovial fluid. Which is secreted by the synovial membrane. This entire joint is surrounded by the joint capsule. Okay, and then we've got a couple muscles and tendons to label. On the back here, we have the triceps, again, responsible for the flexing of the elbow, and it ends with a triceps tendon. Important to note that that connects to the ulna. And wow, I'm really kind of running out of room up here. It is not a very good idea to have crisscross lines in our drawing, so I'm going to have to start writing kind of small. Okay, this is our biceps muscle. And of course, leading into our biceps tendon. Biceps tendon. Okay, so that's an example of a endoskeleton synovial joint. You don't need to necessarily include this in your drawing, so I'm going to draw this in blue here. Insects don't have endoskeletons, they have an exoskeleton, but they do still have muscles. So their muscles are going to attach to the inside of their exoskeletons, something like this. They still, however, work in antagonistic pairs. Okay, so when this muscle contracts, it's going to pull the leg in that direction. And when this muscle contracts, it's going to pull the leg in this direction. So just a little bit of a different setup there. And that's it for the elbow joint. Okay, so speaking of muscles, one of the things that we need to understand about a muscle cell is that it is full of fiber bundles called myofibrils. And myofibrils are made of repeating units called sarcomeres. Okay, sarcomeres on their own have repeating units of proteins called actin and myosin. Okay, so myosin is kind of a thick fiber. Okay, that's in the middle of a sarcomere. Okay, so it's much thicker. And it is going to stay stationary. Okay, once you learn about muscle physiology, you're going to learn that myosin has these little heads which attach to this thinner protein called actin. And those myosin heads cause actin to kind of slide inward. Okay, so when you learn about muscle physiology, you're going to learn that this myosin kind of pulls this actin closer towards the center of this sarcomere. All right, so let's label a couple things on this sarcomere. So first we have the actin. It's important to note that the actin is thinner and it's towards the outside. Okay, on the inside we have this thicker protein called myosin. And these areas where the actin fibers are kind of connected together are called Z lines. And Z lines are important because we can tell a lot about whether a muscle is contracted or relaxed based on the Z lines. So if I'm seeing if I'm looking at a muscle, okay, at a sarcomere in particular, and I see that these Z lines are very close together and that there's a lot of overlap between the actin and the myosin. Okay, that must mean that this myosin has been able to pull that actin. This myosin has pulled this actin closer towards the middle, and this tells me that my muscle is in a contracted state. I know that it's in a relaxed state, okay, when these Z lines are farther apart and there's not as much overlap between the actin and the myosin, okay, because the myosin has allowed the actin to kind of slide off of it. Okay, so when those Z lines are close together, we're looking at a muscle in a contracted state. When they're far apart and we're not seeing much overlap here between the actin and myosin, it is relaxed. Okay. Let's do a diagram of the kidney. All right, when you're starting with a diagram of a kidney, it's important to note that the kidney, okay, is shaped like this. It's got this indented area over here, and that's going to be really important in terms of our kidney function. Okay, this tells me I'm looking at someone's right kidney. Again, as if they were laying down on an operating table. Over here would be the right side and left side. Okay, this portion here is going to point in towards their spine. Okay, so the kidney has a couple of blood vessels going into this portion here. Okay, it doesn't really matter which one you draw on top, okay? But we've got the renal artery and the renal vein. I'm going to go back and label those later. That handles the blood flow. The other thing that exits the kidney is going to be the filtrate or the urine, the materials and water that we are removing from the blood, and that is going to lead away from the kidney via the ureter. Okay, in here on the inside of the kidney, we've got a couple of different areas. Okay, we've got this area called the renal pelvis. Okay, this is going to be responsible for the collection of filtrate, which is going to then exit via the ureter. And then we've got these little lobe type things. Okay, called the medulla. And the rest of the kidney is called the renal cortex. Okay, so time to label this kidney here. Again, this is called the cortex. Each of these little lobes is called the medulla or renal medulla. Renal means kidney, so it's not a bad idea to add renal to each one of these. Again, here's my renal pelvis. The renal pelvis collects filtrate and removes that filtrate through the ureter. And then we've got two blood vessels. We have the renal artery and the renal vein. Okay, so just for your information, you can choose to add this or not. Okay, the renal artery brings oxygenated blood from the heart into the kidney. Okay, the kidney then uses some of that oxygen for its own purposes, and it's also going to filter that blood. About 25% of our blood volume at any given time is flowing into this renal artery. And I guess while I'm at it, I should add a Y to renal artery. Okay, and then the filtered deoxygenated blood is going to leave via the renal vein. Okay, in your studies of the kidney, you should definitely be able to list some differences between the renal artery and the renal vein. Okay, so I can do that for you down here. So kind of separate of our drawing, we can contrast the renal artery, okay, with the renal vein. Okay, they should really have the same amount of glucose and amino acids. Okay, the kidney shouldn't be removing any of those things. Okay, the renal artery is going to be higher in water, higher in salt, and higher in urea, because the job of the kidney is to remove some of those things. Okay, so in general, the renal vein is going to have less water because some water is leaving via your urine. Okay, less salt because salt is leaving via your urine, and less urea because urea is leaving through your urine. Okay, so if I'm thinking about this in kind of a math equation, the renal artery includes all of the things that are in our urine and our renal vein, okay? Because the job of the kidney is to take things out of the blood and remove them via urine, okay? So these have everything, okay? This has everything except for what we removed via the urine. Before we start on the drawing of the nephron, let me just tell you, you do not have to draw a nephron on your IB exam. You may be asked to label it, or you may be asked questions that require your understanding of the structure. Okay, so this is going to get a little bit complicated here, but it'll be worth it. I'm going to start off drawing the tubule and then go back and draw blood vessels later. The tubule begins in this area called Bowman's capsule. Bowman's capsule is going to surround the glomerulus, which we'll get to later. And Bowman's capsule leads into kind of a twisted part of the tubule called the proximal convoluted tubule. Proximal meaning close to Bowman's capsule, convoluted meaning twisted. The proximal convoluted tubule then leads into this long loop called the loop of Henle. Okay, and now's a good time to tell you that this loop of Henle kind of dips down into a different part of the kidney. In this top part of the kidney, we are in the cortex. Okay, so make sure you refer back to your kidney drawing, but this is going to be towards the outside of the kidney. And the loop of Henle dips down into the medulla, which is closer to the inside of the kidney. Okay, so this is kind of more pointing towards the outside of the kidney, and then the inside of the kidney towards the renal pelvis. Okay, this loop of Henle then leads into another convoluted section called the distal convoluted tubule. Okay, distal meaning further away from Bowman's capsule, and then into an area called the collecting duct. Okay, so before I draw the blood vessels, let me go ahead and do some labeling here. This is Bowman's capsule. It surrounds the glomerulus. Any filtrate that ends up in Bowman's capsule leads into the proximal convoluted tubule, which leads into the loop of Henle, and then into the distal convoluted tubule. And then any filtrate left in the convoluted tubule is going to move into the collecting duct. And that collecting duct is eventually going to end up into the renal pelvis and then into the ureter. Now it's time to add in our blood vessels. Okay, blood is going to enter Bowman's capsule via the afferent arteriole. That's going to be unfiltered blood. It's going to come in here and go through a series of squigglies known as the glomerulus. The glomerulus is kind of a hot mess, tangled bit of capillary. The glomerulus has a bunch of fenestrations or holes, okay, in this mess here that are going to allow a bunch of blood solutes and liquids to be filtered out. Blood is going to then exit, okay, the glomerulus via the efferent arteriole. All right, now go back and check your drawing. Make sure that your afferent arteriole is wider than the efferent arteriole. What happens is blood is going to enter through the afferent arteriole, and because that's wider, okay, it kind of gets funneled in here, and then when blood leaves through a hole that is smaller than the hole that it enters through, okay, what that's going to do is create a lot of pressure in here. Kind of like if you think about a funnel having a wide opening and a narrow exit, okay, that's going to create a lot of pressure in here, which is going to force solutes out of the glomerulus into Bowman's capsule and then in through the rest of our nephron. Now, this process is called ultrafiltration, and it removes way too many solutes, okay? So these solutes and some of these liquids, like the water, need to return to the blood. So this efferent arteriole is going to kind of loop back around. Hopefully you're doing a better job of drawing than I am. Okay, over here, and it is going to form a capillary bed. And that capillary bed is going to kind of hard to explain here, but it's going to branch out and kind of hit a bunch of different parts in our nephron. And it's got other overarching branches. Now our diagram is kind of becoming a huge mess. Okay, but that's okay. Okay, and then now, okay, what's going to happen, this ultrafiltered blood, okay, is going to lose a bunch of solutes and filtrate to Bowman's capsule. That blood is then going to come back around and pick up materials that we can selectively reabsorb from the proximal convoluted tubule, from the loop of Henle, and from the distal convoluted tubule. Okay, so anything that is reabsorbed, okay, by the bloodstream is going to make it back into this blood here, and this blood is going to go to the renal vein. Okay, time to label some of these blood vessels. Okay, this again is called the afferent, that's an A, The afferent arteriole brings in blood, unfiltered blood, from the renal artery. So I don't know if you want to make a note of that. I'm going to try to write that really small. Unfiltered blood from, I'm going to abbreviate here, the renal artery. Okay? Blood is going to then go through this structure called the glomerulus. It is going to exit through the efferent arteriole. Okay, I remember E for efferent, E for exit. Okay, so it's exiting Bowman's capsule. Okay, it's going to go, blood is going to exit Bowman's capsule, okay, exit the glomerulus, go through the efferent arteriole, and then pick up things that are selectively reabsorbed by these tubules. Okay, and this network of capillaries is called the peritubular. Peri meaning around, tubular meaning around these tubules, like the proximal convoluted tubule. The peritubular capillary bed, or peritubular capillary network. Okay, and then this blood is going to exit, and this is going to be filtered, because we just filtered it with the nephron, filtered blood to the renal vein. Okay, and if you're wondering what's going to happen to the filtrate, the things that weren't reabsorbed by the blood, this filtrate is going to go to the renal pelvis, where it will be collected and then passed via urine into the ureter. And that is the nephron. Okay, now remember, the function of the proximal convoluted tubule, okay, is to selectively reabsorb some of that stuff that we took out of the blood during ultrafiltration in Bowman's capsule and in the glomerulus. So we took too much out, we got to put some stuff back in. If I were to take a cross section of the proximal convoluted tubule, okay, I'm going to notice that it's kind of a circular structure. Okay, made up of many cells. Okay, so something kind of like this. So I'm looking at this cross section, kind of like taking a slice out of a piece of bread. I'm looking at a slice of the middle of this proximal convoluted tubule. And it's got several structures that are going to allow it to do its job, which again is to take things from the filtrate and move it back into the bloodstream. So here we have the lumen, and the lumen is where the filtrate is. Our job is to get things from the filtrate back out of that tubule, because out here is our peritubular capillary bed, and we need to take some of the stuff that we took out of the blood and put it back into the bloodstream. Okay, so these cells are all eukaryotic, so they have a nucleus, which is fine. We don't really need to label that because that's not a specific structure that is going to really help it do its job here. Okay, a couple of things that we should know. These cells are lined with microvilli. And just like the small intestine, these microvilli are going to increase surface area for reabsorption. Okay, so it works the same way as the small intestine, increasing surface area for reabsorption. You do need to know the function of those. Inside of each of these cells are tons of these organelles that I'm doing a really bad drawing of, and these are mitochondria. Okay, so again, remember what the job of these proximal convoluted tubule cells are. They need to take things that are in the filtrate. We took too much out of the blood, it's in the filtrate, we need to get it back into the bloodstream. Remember, there are capillaries all over the place here. You don't need to draw them, okay? We need to get materials from the filtrate into these capillaries. So the microvilli and the mitochondria have some important functions in doing that. The microvilli are going to increase surface area for reabsorption. And then these mitochondria are going to generate ATP for active transport. Some of the things that are in the filtrate, like salt ions in some places, okay, need to go back into the bloodstream. But because the concentration in the bloodstream is already higher than in the cells or in the filtrate, these mitochondria are going to be called on to generate ATP so that they can be pumped from the filtrate and then into the bloodstream. Okay, and so that is the structure and function of the cells lining the proximal convoluted tubule. Okay, the last drawing of the kidney involves the effect of this hormone called the antidiuretic hormone. Let's go ahead and write that out. Antidiuretic, it's an R, hormone. The antidiuretic hormone is produced by the posterior lobe of the pituitary gland, located near the hypothalamus, deep within your brain. When you study hormone production, you'll need to go back and figure out how this posterior pituitary works. But basically, it goes something like this. The hypothalamus has neurosecretory cells that empty into the posterior pituitary. So antidiuretic hormone is actually produced in the hypothalamus but secreted by the posterior pituitary. And the antidiuretic hormone acts on the collecting duct. Okay, so the collecting duct is part of the nephron. Okay, so I'm going to draw two different drawings. Okay, and it's going to look a little something like this. If ADH is present, and if ADH is not present. Remember, it's a hormone, so hormones are going to be circulating in your bloodstream. So when I'm talking about present and non-present, I mean in your bloodstream. Okay, so let's draw what's kind of happening here. Here's my distal convoluted tubule and the collecting duct. So I'm going to abbreviate this: distal convoluted tubule and collecting duct. Okay. If ADH is present, it allows this collecting duct to become permeable to water. So what that means, that says H2O. Okay, what that means is that water is going to exit the collecting duct and it's going to return to the blood. Okay, remember that out here are a bunch of capillaries. Okay, now that means that the water is going to stay in the blood or I should say more water, stays in the blood, okay, and there is less water in the urine. Okay, because remember, what's happening here is anything that makes it through the collecting duct is going to become urine. So if we're removing water from the collecting duct, that means not as much is going to make it into the urine, more is going to go back into the blood. So your posterior pituitary is going to produce ADH when it needs to conserve water. Okay, when it needs more water to stay in the blood, it's going to release ADH, which causes this to become more permeable to water, meaning more water is going to stay in the blood. So like if you're sweating and you're already losing quite a bit of water, or if you are dehydrated, okay, your posterior pituitary will secrete ADH, okay, to make sure that water stays in your blood and isn't passed as urine. On the other hand, if we are already well hydrated or we're drinking too much water and we need to regulate the amount of water in our bloodstream, then the posterior pituitary will choose not to secrete ADH. This collecting duct becomes impermeable to water, okay, and water is going to leave via urine. Okay, so the water stays in the filtrate or urine, and it doesn't return to the blood. Okay, so again, when you already have enough water in your bloodstream, okay, and you can't maintain those levels of water because we need our blood plasma to be isotonic to our blood cells, and we need to get rid of some of that extra water, the posterior pituitary will stop producing ADH. This collecting duct becomes impermeable to water. That water leaves as urine, okay, and does not return to the bloodstream out here. Spermatogenesis is the process of generating or making sperm. It occurs in the testes and more specifically inside of the seminiferous tubule. Okay, so spermatogenesis starts with the spermatogonium, which is a diploid cell, and it can either undergo mitosis to replace other spermatogonium or meiosis, which we're going to focus on since we are talking about the generation of gametes. Okay, so after meiosis starts, we're going to have, again, meiosis 1, and that's going to result in 2 cells. Now, since there is a copying of the genetic material, each of these is still going to have 46 chromosomes, but because of the way that meiosis 1 works, okay, those 46 chromosomes are no longer in homologous pairs, they're in sister chromatids, so we say that they are haploid at this point. And these are what we call primary spermatocytes. Okay, now each of these is going to undergo meiosis 2. And that's going to finally reduce that chromosome number down to 23 in humans. And these are called secondary spermatocytes. Okay, now each one of these has the capability to develop into a sperm cell or spermatozoa. And these, of course, would then travel to the Sertoli cells, or rather attach to the Sertoli cells inside the seminiferous tubules to gain nutrients so that they can differentiate and become fully functional sperm cells. Okay, so speaking of spermatogenesis, again, that happens inside of the testes in these structures called seminiferous tubules. Okay, so if we were looking at a testy, and let's see, inside would just be this network of tubules. Okay, if I were then to take one of these tubules and do a cross section, it would look something like this. Okay, so on the outside here is the perimeter. Of the seminiferous tubule. Okay, inside of the seminiferous tubule is going to be a collection of some rather large cells, probably more than 4, but I'm just going to draw 4 for right now. And these large cells are called Sertoli cells. And Sertoli cells are there to provide nutrients for the developing sperm. Okay, inside or kind of around those Sertoli cells are our spermatogonium, and we talked about those in the last video. Spermatogonium are those cells that produce sperm. So pretty neat cells that can either go or undergo mitosis or meiosis. Okay, inside here is what we call the lumen. That's just a general word that we have for the inside of a tube. And attached to the Sertoli cells are the developing sperm. Or you may hear that or see that written as spermatozoa. So these developing sperm are going to gain nutrients from these Sertoli cells. Okay, when they're done doing that, then they're going to be moved to the epididymis. Okay, so what does a sperm cell look like once it's done differentiating? All right, well, it's pretty simple. We're going to start out with a haploid nucleus. And kind of in front of that nucleus is a little sac of enzymes called the acrosome. Right behind that is a set of organelles. Sperm cells really don't have very many organelles. It's pretty much limited to just these mitochondria. Okay, and those mitochondria are of course there to facilitate the generation of ATP so that it can move this structure around, and this is called the flagella. And all of this is contained within the cell membrane. Okay, so let's go through and label this. Here is our haploid nucleus. Okay, this is the acrosome. And again, this contains enzymes needed for fertilization. More on that another time. Here are our mitochondria. And these guys generate ATP. And in this case, it's for movement. Okay, here is the flagella. And again, that's for movement. Sperm have a long way to swim all the way from the vaginal canal through the cervix, up through the uterus, and into the fallopian tube. So they're definitely going to need this. And all of this is surrounded by the cell membrane. Oogenesis has some things in common with spermatogenesis. They're both forms of meiosis. They both produce gametes. Okay, but spermatogenesis happens on a daily basis in men, whereas in oogenesis, this is really only going to happen shortly after the time of birth. Okay, so oogenesis is going to start with a diploid cell called an oogonium. It's present in the ovary. And the oogonium is going to undergo meiosis 1, just like spermatogenesis. Okay, but this is a little bit different. One of the cells produced is going to be much bigger than the other one. Okay, they're both going to be haploid, again, because they're no longer in homologous pairs, but all of the organelles present in this oogonium, like the mitochondria, the Golgi, the endoplasmic reticulum, the centrioles, is going to get passed to this product of meiosis 1. This is called a polar body, and polar bodies are basically just dumping ground, okay, for those extra chromosomes that we don't need. Now, at this point, this is what we call a primary oocyte. Okay, and these are going to be present in the ovaries. Once a month, one of these primary oocytes is going to be kind of chosen to go through the ovulation cycle. Okay, and the ovulation cycle is then going to, how do I say this? Kind of stay arrested in meiosis 1 until fertilization. So let's make it clear that this next step does not happen unless this primary oocyte is fertilized by a sperm. So let's assume that we have a sperm. Okay, again, these are just kind of dumping grounds, okay, for chromosomes. They're not going to become viable. Okay, they're going to die off. Same thing here. Okay, so just like last time, one of those cells is going to retain all of the organelles and it's going to stay very large. The other one is going to just be a dumping ground for those extra chromosomes. And this is what we call a secondary oocyte. Okay, but it's not a secondary oocyte for long because remember, this step doesn't happen unless it's fertilized by a sperm. So as soon as this turns into a secondary oocyte, okay, a sperm will have made its way all the way in here at this point, and the sperm nucleus and this nucleus will fuse to make a zygote. Okay, so let's take a closer look at this secondary oocyte. Okay, we're going to have a few features here. Again, we have our secondary oocyte in here. Okay, there's a cell membrane, there's a bunch of stuff in the middle that we'll get to. And then surrounding it are some layers. We'll only draw one layer, okay, of follicle cells. So these were on the primary oocyte as soon as it was formed, and they stay on them in the ovary. During the ovarian cycle, a second layer would develop into the Graafian follicle. But remember, the Graafian follicle stays behind in the ovary, and the follicle cells hang on to that oocyte during ovulation. All right, so let's go through and do some labeling here. Here is our follicle cell layer. Okay. This area in here is called the zona pellucida. Okay, its function is to facilitate what's called the cortical reaction. Okay, I'll go over that at the end of this drawing. Okay, here we have the cell membrane of the ovum, which is the egg. Another way of saying that is secondary oocyte. It means the same thing. Okay, and inside of here, we're going to have a few things. Okay, here's our haploid nucleus that will eventually fuse with the sperm's nucleus to form a zygote. Also in here is a yolk. Yep, just like a chicken egg. And this is going to serve as the energy reserves for when this zygote, should it get fertilized, when the zygote needs energy to undergo mitosis. Also in here is a full set of organelles. Okay, so kind of all of them. The Golgi, the ribosomes, the mitochondria, the lysosomes, the centrioles, all of them. Okay, and also inside of the ovum, it's going to be what we call some cortical granules. Okay, now the cortical granules and the zona pellucida have a linked function. When a sperm penetrates the follicle layers, and that might happen, many sperm might penetrate this layer, okay? When one of the sperm's cell membrane fuses with the ovum's cell membrane, that's going to trigger these cortical granules to cause the zona pellucida to harden. Okay, so this becomes hardened, meaning that every sperm that's still out here is not going to be able to penetrate the egg. Okay, so before we start on drawing the blastocyst, we should probably talk about a couple of things with the blastocyst. It's about 100 cells, okay, in size. But I say size loosely because it's pretty much the same size as the ovum. So think about normal cell division. Normal cell division, okay, we have one cell becoming two cells that are roughly the same size, okay, and that would add to the overall size of the cell mass. It would become bigger and bigger. But in early embryonic development, that one zygote just divides into many cells without becoming any larger. So imagine taking a whole pizza, and cutting it up into 100 slices. The pizza doesn't get bigger, it's just in 100 parts. Now, the blastocyst, as it's developing into these 100 cells, is kind of hungry. It needs a lot of energy to do all of that mitosis. And it's using energy from the yolk, but eventually that energy is going to run out. And the blastocyst is going to implant itself into the endometrium of the uterus. Okay, now remember the endometrium is the highly vascular lining in the uterus. And the blastocyst kind of looks something like this. Okay, so we have a layer of cells around the outside. Again, these aren't follicle cells anymore. We're done with that. These are totally different. And then on the inside, most of this is just going to be fluid, but then kind of tucked up close to where the endometrium is what we call the inner cell mass. Okay, so something like that. So the inner cell mass, okay, is eventually going to become the embryo. Okay, we have a fluid-filled cavity, and then we have what's called the trophoblast. And the trophoblast, here's how I remember this. This word tropho, we see that a lot. Autotroph, heterotroph, trophic level, having to do with eating. Okay, well, these trophoblast layers eventually become the placenta, which is how I kind of think of a fetus as being able to eat. Okay, so the trophoblast, those cells become the placenta. And again, this whole structure is known as a blastocyst. Okay, so once that trophoblast becomes the placenta, Okay, we can start to understand how materials are exchanged between a fetus and its mother. All right, so here's my really bad drawing of a fetus. I say really bad, I don't know. It's not that far off. Okay, and extending from the fetus is, of course, the umbilical cord. Okay, and the umbilical cord has 3 blood vessels. Okay, so 1, 2, 3. They don't look branched like this. In reality, there's kind of a big, if that's actually a vein that carries oxygenated blood in a fetus. We'll talk about why in a minute. And then we have kind of like 2 fetal arteries kind of wrapping around it, and that's all in this umbilical cord. Okay, so it's going to look something like that. You don't have to draw that part. I just wanted to kind of give you an idea of what's going on there. Umbilical cord. Okay, so here we're going to be taking blood to the mother or to the placenta. Okay, and that's going to be going through that blood vessel. Blood is going to be going to the placenta in this blood vessel also. And then this blood vessel is going to carry blood from the placenta. So going this way. Okay, so if we're thinking about things on like an oxygenated or deoxygenated level, okay, if it's going to the placenta, okay, it's not going to the placenta to donate anything. It's going to the placenta because there are waste products in there or because it needs oxygen. So this is full of deoxygenated blood. Same thing with this one. The blood coming from the placenta is free of waste products. It has oxygen, okay, all of the things that that growing fetus might need. Okay, now these blood vessels, I should have drawn these a little bit longer. Okay, these blood vessels are going to kind of end in a network of capillaries. Okay, so capillary network, capillary network. So those are our fetal capillaries. Okay, those fetal capillaries kind of back right up next to maternal capillaries. Okay, and those maternal capillaries are, of course, connected to maternal blood vessels. So here we're carrying blood to the placenta. Okay, and over here I guess we should make a note that this is the mother side. I'm not going to draw her. Okay, and down here would be the blood from the placenta. So if it's going to the placenta, it's going to carry oxygen and other nutrients. Okay, if it's going this way. And if it's coming this way, okay, it's carrying deoxygenated blood. Okay, full of waste products, etc. Okay, so just to give you a quick rundown here. Okay, the mother is going to donate energy-rich, nutrient-rich, oxygenated blood to the placenta. Okay, those hormones, nutrients, oxygenated hemoglobin molecules are going to diffuse from this capillary network to this capillary network and go into the fetus. The fetus is going to use the oxygen, going to use the nutrients, okay? And it's going to produce blood that is high in waste and low in oxygen. It's going to send that blood to the placenta where it makes that exchange again. And again, it's this network of capillaries that we call the placenta. The placenta is just that exchange place for those fetal and maternal blood components. Okay, our last drawing that we're going to do for chapter 11 is a graph of hormones throughout pregnancy. Okay, so on the x-axis here are our days of gestation. Okay, so here we're at day 0. Birth usually occurs just a little bit before 280 days. So that would put halfway at 140. Okay, and then 210 and 70. And I'm going to be graphing 2 different hormones. I'm going to first start out with progesterone. Okay, good luck reading that. Okay, progesterone is at first going to be produced by the corpus luteum. Okay, eventually that corpus luteum is going to stop producing the progesterone, and it's going to be the placenta that produces that progesterone. Okay, next one I want to produce is estrogen. Estrogen is also produced by the placenta, but it doesn't really start until the second trimester. So it's going to kind of start down here and go up more in a linear fashion. Right before birth, so when the fetus is mature, both estrogen and progesterone are going to severely plummet. Okay, this plummeting in estrogen and progesterone is going to initiate the birthing process. And of course, you know that you're going to need to know things like oxytocin and after birth prolactin, but we're not going to cover that with any drawings. So that'll do it for chapter 11.